Alright, class, now we're going to be talking about proteins. Proteins and nucleic acids are two other macromolecules. So proteins are just big old long chains of amino acids. And so depending on where these amino acids are in the protein, it will determine the protein shape and specific functions. Every amino acid is made up of an acid, which is carboxyl, amino group, and an R group. That R group is how they are different. And they all have CHON, carbon, hydrogen, oxygen, and nitrogen. They have a huge amount of functions. Structural things are keratin for hair and nail, collagen, all your ligaments, tendons, skin. Metabolic processes, enzymes. This is a huge part, so huge that we're actually going to have a separate PowerPoint about it. And what they do is catalyze reactions, which really means they speed up reactions. Without enzymes, pretty much nothing would happen in your body, but you can think of digestion, everything that breaks down in your body, enzymes are a part of, and there's a specific enzyme for each little tiny function in your body. Other things they do is transport things across your cell membrane. So later when we talk about cell membranes in our next unit, we'll go into the different proteins that help different things go across. Another important part is hemoglobin, which is part of your blood. Hormones are also a type of proteins, such as insulin. An important characteristic of proteins is they can be called denatured, which basically means that heat will cause them to lose shape and functionality. And like I said, more on this later. And so these peptide bonds, this is what is found in a protein, is just a fancy name for proteins when amino acids are being together. So we also did this in class, and we just are taking out water. It's a weird way of showing it, but H2O. And so again, dehydration, took out water, and when they link together, it's the peptide bond. And another word for protein is polypeptide. So just like in carbohydrates, how we had polysaccharides, now we have polypeptides. It just means many amino acids or many protein. There are different shapes. Remember we said depending on which amino acids were present would determine the shape and function of the protein. So the first thing is called the primary structure. So primary is 1, and all it is like this picture, a sequence of amino acids in a line. And so it's just a line, a linear form, and they form a polypeptide chain. The secondary structure, which is shown in both of these pictures, are when there's certain amino acids that are attracted to each other, and so they start folding into coils and folds. And so kind of this helical alpha helix or this pleated sheet. And this results from weak hydrogen bonds and the stiffness of the peptide bonds. And so there's going to be some stiff parts, and the hydrogen bonds then break in order to form this structure. After that, we get tertiary. And so these are just like some weird contortions, and they start bonding the side chains, which are the R groups. And some of them are hydrophobic, and so they want to get away from the water, so they go inside. And then there's the hydrophilic, and they don't mind being outside. The last one is quaternary for 4. And really all this is 2 or more polypeptide chains or subunits. An important word to remember this is globular. So it's just putting a couple of these like tertiary things put together. So you see hemoglobin here, and this is collagen. Here's another, just review, primary structure. So one old big line of amino acids. Secondary, you have your helixes and pleated sheets. Tertiary, now we're getting some weird contortions just due to ones that want to be inside or around. And quaternary, just get 2 of those and put them together. And so I have a little piece I'll show you more about this. And so this is just a little animation that shows it as well. So this long piece, this is primary structure. And what happens, and so then it just folds up, and that's basically what's happening with protein folding. So going in more in depth, we have the secondary structure, alpha helix, or the beta sheet, which is also the pleated sheet. And it's just showing the bonds going in there. Sorry, those are the alpha helixes. Now this is showing more of the beta sheet, the same parts. This is what, with computer modeling, what an actual protein looks like. Sorry, I don't know what that is, so I'm gonna close that out. This is going into more stuff than you guys need to know about, but this is the actual protein, what's going on. Let me actually go over to here. So the protein folding, you get your primary structure, and then it's going to fold. And so this is the alpha helix, this is the beta sheet, how they're representing them anyway. Another important thing about proteins is that they don't always fold right, and they may not always be made correctly. And so if it folds, it's not going to work. That's going to go into the lysosome and get destroyed, or it can go to the correct place. And so let's say this is our secondary structure. It's happy. Now we want to go to the tertiary, which the tertiary, and so the folding due to hydrophobic and doing different things. And then you go to quaternary, you're just adding the two together. And you get your quaternary structure. And this kind of shows you an overview of everything. Primary, secondary starting to fold, tertiary, and the quaternary. And we're going to go back to alright. The last one we're going to talk about is nucleic acids. And so again, nucleic acids are DNA. They're going to store and transmit hereditary information. They also aid in protein synthesis, which is just how proteins are made. The two examples of them are DNA and RNA. DNA, the long name for that is deoxyribonucleic acid, and RNA is ribonucleic acid. There's two types of bases, pyrimidines, which are T and C, and purines, A and G. We'll talk more about that when we go into genetics, but for right now, you just need to know the words pyrimidines and purines are nucleic acids. The structure of DNA, which are nucleotides. Nucleotides are what make up the nucleic acid. The different parts, there's 3 different parts: the 5-carbon sugar, nitrogen base, AT, G, and C, and phosphate group. Another just important part is that A always pairs with T, G always pairs with C. And so if you look, C's and G's are kind of rounded. That's how I remember they go together. This is an actual folded out part, close-up look of DNA. And so this is T, A, G, and C. So these are the base pairs, the bonding in between them. And notice that each one of them is connected to a sugar and a phosphate group. And so that is what we call the backbone of DNA. And these are the rungs, the middle part of the DNA. Alright, to put everything all together, I'm going to go through this. Is a really important piece to make sure you guys are writing down, because this is going to put everything we talked about in these last 2 PowerPoints together. I have a lot of test questions on this. Alright, so the 4 macromolecules. We have carbohydrates, lipids, protein, and nucleic acids. And then we need to know what the subunit, another word for that could be building block, another word is monomer. Mono, one, mer, molecule. And so what did we start with all of these? Carbohydrate, we have sugar, or technically glucose, lipids, you have fatty acids, protein, amino acid, and nucleic acids, the nucleotide. So you take all of these and you keep building up, and that's how you get your bigger molecules. What elements do they have? Carbohydrates have CHO, and it's in that 1 to 2 to 1 ratio. Lipids have CH and very little O, so 1 to 2 and just a little bit of oxygen. Carbohydrates, we add that amino group, which has nitrogen in it, so we have CHON. Nucleic acids have that, but again, they have that phosphate group as the background, so they have CHOMP. And that is it for the macromolecules. Look forward to enzymes.